Սիրելի ուսանողներ, այսօր մենք մեծ պատվի ենք արժանացել այս բեմահարթակը համալսարանը վերջին տարիներին Նոբելյան մրցանակի դափնեկիրների տարեկան ավանդույթ ունի, բայց այսօր առանձնահատուկ օր է այս համալսարանի համար, որտեղ այս համալսարանում այսօր իր զեկույցով հանդես կգա մեր ազգի առաջի Նոբելյան մրցանակի դափնեկիր Ardem Patapoutian-ը։ Բարի գալուս ես։ Ես կուզեի ձեզ տեղեկացնել, որ րոպեներ առաջ այն կուրսի հատիկը, որի վրայից դոկտոր Patapoutian-ը ստացել է հայտնի մոլեկուլը, այսօր տնկվեց նաև բժշկական համալսարանում և մենք սպասենք այդ գիտության ծառի տուներին, հաշվի առնելով, որ մի քանի րոպե առաջ նաև դոկտոր Patapoutian դառավ COBRAIN ծրագրի advisory board-ի անդամ, 7 անդամներից 2-ը Նոբելյան մրցանակի դափնեկիր են։ Այս համալսարանը կտրուկ բերել գաղթում գիտական ասպարեզում և Գիտական խորհրդի որոշումով նոմինացիոն կոմիտե ենք դիմել, որ այն պատվարժան շարքում, որտեղ որ մեր պատվավոր դոկտորները իրենց ուրույն դեմ են գտել նաև առաջի հայազգի Նոբելյան մրցանակի դափնեկիրը ևս ընտրելի Երևանի պետական բժշկական համալսարանի պատվավոր դոկտոր։ Համեցեք ընտրելուց։
So I know some of you are, experts in molecular biology and other forms of biology, but some of you maybe have a little bit less experience in this. So I prepared a talk that I hope will learn something. Either you're an expert or have a high school degree. So let's see how this goes. Welcome, and thank you for having me and for the tour and the very nice introduction. So, I think our sense of reality is really based on our senses. And over the last few decades, scientists have found at the molecular level how we see, smell, and taste. And yet this one in red, the sense of touch, has been more enigmatic. So the sense of touch gathers information from our environment related to temperature, pressure, even chemicals, and translates this to the brain into language that it understands. However, at the molecular level, how do we sense our environment was not very well understood. Beyond this sense of touch and pain, most of the cells in your body communicate through chemicals, and again, we have learned a lot about these kind of signals. But cells also experience mechanical forces, pressure, and we know much less about this. But I'm here to tell you that your sense of touch is quite amazing. So human tactile discriminatory ability extends into the nanoscale. So you can, with your fingers, discriminate something in the diameter 1/500th of a human hair. So that's quite an amazing ability. On top of that, we rely on effective touch for our emotional health. And we all knew a lot about this during COVID, where we had to stay separated from each other. And from the related sense of proprioception, you know where your limbs are in space without looking at them. Because of this sense that we'll talk more about in a little can stand up, walk, or, as demonstrated by this picture, do complex tasks, such as playing a musical instrument without even looking at your hands. But I show this picture because I think it's, for me, the photograph that illustrates the power of touch more than any other photo. So this is, of course, President Obama in the White House, and this African-American little boy is visiting the White House. And the first thing he tells him is that his friends tell him that his hair has similar texture and look to the Obamas. And President Obama immediately bends over and says, "Why don't you touch it?" And of course, we're not just talking about sensing the texture, but more of the intimate, affective nature of touch that's so important for humans. So we often say, seeing is believing, but President Obama knew that touching is also believing. So the flip side of touch is, of course, pain. And acute pain is absolutely essential for our survival. It's a warning signal of damaging stimuli being in our environment. But chronic pain, which is really defined as pain when the stimulus that initially caused it is no longer there, but we still feel pain, is a major unmet medical need in our society. So how do we sense all of these stimuli that I talked about? The gatekeeper for all these sensors is a group of neurons that live just above your spinal cord, called dorsal ganglia. And they have very specialized, heterogeneous group of neurons. So some only sense temperature, pain, light touch, or this enigmatic sixth sense that I talked about, proprioception. So what do? They go all over the body in the periphery and innervate different types of cells and gather information. So for example, in red, you can see these proprioceptive neurons actually wrap around muscles, and from how much your muscles stretch can tell you where your limbs are in space. Others innervate in the skin and help you sense either changes in temperature or pressure as experienced by touch, for example. In addition, we have some of these neurons even innervate internal organs, and we'll talk about this. We call this interoception. So for example, you can feel when your bladder is full. I wanted to show this video from a zebrafish, a type of fish developing, where it's been videotaped while these neurons, you see them like little grapes on this, developing and extending their processes into the body. As you can see, they're beautiful, and they send these axons all over its body just to help it sense its environment. But what we're really interested in is more at the molecular level. How do you sense changes in temperature and pressure? So in other words, how do you sense a gentle breeze or a hammer hitting your finger? So it was known for decades that there are specialized molecules, proteins called ion channels, that get activated specifically when temperature or pressure changes. So over the last 25 years, David Julius's lab, my lab, and others identified some of these ion channels involved in temperature sensing. I will not be talking about this today, but how mechanical force, pressure is sensed was not known, and this is what I will talk about. First, just give you a couple of very introductory slides on what these molecules are. So we call them ion channels. They transmit ions across the plasma membrane. So all of your cells, for its protection, is surrounded by a lipid bilayer, a fatty element that protects the cell from the outside world. But for cells to be able to communicate with the outside world, they need specialized proteins. And these proteins shown here, for example, in multicolor, sit in the middle of the lipid bilayer shown by these yellow markings. And they are very simple machines. They're either closed, don't do anything, or they're open, and ions will flow into the cell, which is the signal that neurons know how to recognize. They are fantastic biological machines. Once open, a single molecule ion channel can let about 10 million ions through per second. And when these ions flow, it actually changes the voltage across the membrane, just like electricity. And when these electrical impulses flow, neurons get activated. But these machines are extremely microscopic. So just to give you an idea of how big they are, a grain of rice is about 500 times bigger than a cell. And if we amplify a cell, this is what it looks like, for example. It's about 10 micrometers in diameter. And ion channels will be sitting at the edge of these cells, so small that you can't even see, even with a light microscopy. So we're looking for these machines that transform pressure into chemical signal, but they're very, very difficult to see. So my partner in my lab, the postdoctoral fellow called Bertrand Coste, he's French, now he's back actually, he's a professor in Marseille. About 12 years ago he came to my lab to find these sensors. So how would you even look for them? So one of the ways that Bertrand figured is because when you activate them it causes an electrical signal. He came up with this assay where he would impale the cell so he can record electrical currents from it. And as the red arrow shows, with a blunt glass probe, he actually pushes on the cell while he records the currents that go through it. This is a video of what it looks like. So you can see the probe pushing on the cell, and on the electrode that's on my left side, records the currents. And so if this cell has one of these ion channels that I was talking about, you will be able to record their current. Okay, so we know how to record from them, but how did we identify them? So in science, we often talk about a reductionist approach, which means what's the simplest system you can use to answer the question that you want to answer. So instead of going and trying to look at it in the gene neurons that I talked about, he decided that since none of these pressure sensors are known, why don't we find a very, very simple cell that can grow in a dish and find it there, and then we look at it in what we call in vivo in the animals to see what they do. So he found that these cell lines that neuroscientists have worked with for decades in Neuro 2A have robust currents. So we knew that this cell then expresses these ion channels. I won't go into details, but he gathered the number of genes that could potentially express this protein. There are a few hundred candidates, and he proceeded to knock them out, which means to delete them one by one to see which one is required for this current. So this is a simple data slide. What you're looking at in the middle is the maximal, on the x-axis, is the maximal mechanically activated currents recorded by Bertrand. And on the y-axis, each is a simple candidate. So you knock down one, and you see if this current is still there or not. So as you can see, he had lots of negative data. From number 1 to 71, he knocked them down and nothing happened. This took him a whole year to get no positive data at all. But then you can see the happy day, the red one, the 72nd candidate. When he knocked it out, he saw that, and that's a logarithmic scale on the x-axis, we saw a massive decrease in the mechanically activated current. And he knew that he had identified something special. So because most of our genome, the genes, we don't know what they do yet, the name of this gene that he found was called FAM38A, which simply means unknown family 38 member A. He quickly renamed it piezo1 from Greek piezi, meaning pressure. So this slide shows that if you get rid of this one, the expression of this one gene, you don't get this current. But biologists would feel very good if you had the opposite experiment as well, which means you take this gene and express it in a cell that's usually not mechanosensitive and see if it becomes mechanosensitive. And indeed it becomes. This is one of these experiments showing that. This just shows on the left that the more you push the cell, the larger the currents. Those are the recordings in the bottom you can see. And on the right is what we call the quantitation of this. Control is the cell that does not have this channel. There's no currents, but if he expresses this, he sees massive mechanically activated currents. Interestingly, you can look in the genome and see if there are other proteins that look like piezo1. Indeed, there was one other, he named it piezo2. This also, if you overexpress it in cells, you get very large mechanically activated currents. And the two, if you look at what we call the sequence, which puts the protein together, they're quite identical. They're almost 40% identical. So what does this protein look like? Initially, all you can do is look at the sequence and try to guess what it will look like. And the only thing we could see is, I told you ion channels sit in this fatty layer of the cell. To do this, they have to have domains that cross the membrane. So most ion channels cross it twice or maybe 6 times. So piezo was very special because it crosses it 38 times. That's more than any other protein in our genome. So later on, we and others actually solved the structure of the protein. That means that in 3 dimension, we know now exactly what the protein looks like using cryo-EM microscopy. And it's a fantastic looking molecule. Each of these 38 transmembrane domain containing proteins come together as a trimer, which means there are 3 identical units come together. In the middle, they come together to form the pore that you can't really see. Each unit is a different color. Each of these helices that you see is one transmembrane domain. One of the interesting things you'll see about it is that the whole protein is kind of bent, which is very unusual. It kind of bends the membrane in the opposite direction that it normally wants to be. And this is just a rendition of what we think piezos look like in this fatty layer, which is in blue, and you can see in this like purple color that piezos almost dig themselves in a little hole in the plasma membrane. So the idea, this is just a hypothesis still, of why these are pressure sensing machines, is that when the membrane gets stretched, the protein actually straightens and causes the opening of the ion channel. So one of my trainees that started his own lab in Beijing actually recently saw this structure in its flat form, and this video is just going to show you morphing of the 2 structures. I think it's a thing of beauty. So this is the structure you already saw, and the idea is that if you put pressure on it, it actually flattens. And this opening causes the ions to flow into the cell. This is all I'm going to say about what the protein looks like, but most of the work in my laboratory has focused on what do these proteins do in our bodies. And to do this, we study this ion channel using both clinical data to see what mutations in these genes cause, as well as in animal models. And loss of function and gain of function just means, and this is what we do usually to understand function, is what happens if you get rid of this gene, and what happens if you have too much activity with these genes. Those 2 questions tell us what kind of roles they play in physiology and in disease. So this image, which is the RG neurons that I talked to you about, are showing where piezo2, the sister molecule, is what we call expressed. So every cell in your body has all the 20,000 or so genes that you have, but a certain cell only expresses, which makes RNA, about half or less of these, and those are the ones that are functional in those cells. So in brown color shows that piezo2 is very strongly expressed in many of these dorsal ganglia neurons that we were very interested in. So over the years, I'll just give you now a summary. We actually found out that piezo2 is indeed the molecule that helped us to sense touch. And the first, we did these experiments in mice. So if you make a mouse where piezo2 is deleted, and how do we ask the mouse if they sense touch? This was actually not known and very difficult to do, but my students came up with very simple but almost amusing type of experiments to ask this question. In one case, they put a white tape on the back of the mouse and just observe what they do. And here's a normal wild-type mouse. If you do this, they start scratching it. They don't like it, they feel it, and they try to get rid of it. If you take a mouse that doesn't have piezo2 and do the same experiment, they just don't care. They don't even feel it. They don't sense it. So we've looked at this, of course, very quantitatively, and this slide just shows how strong the deficit in touch sensation is. So on the left, you're looking at, you're touching their paws with a little thin hairs and seeing when they respond with their paw lifting. And the wild-type normal animals is in gray. On the x-axis, it's increasing amount of force. As you can see, the more force you apply, the quicker and the more they lift their paws. But without piezo2, in the pink color, you see they don't respond at all. The other two are related assays to assess the sense of touch. So I want to come back to this sixth sense of proprioception that I mentioned. And the reason I like to talk about it is because I think it's actually perhaps your most important sense, but most of you have, I'm sure, never heard of it. So what is proprioception? As mentioned, it is the sense that allows you to know where your limbs are in space. So this is how I can close my eyes and touch my nose. If you think about it, how was I able to do that without looking at my fingers? So some of these sensory neurons actually innervate your muscle instead of skin involved in touch. And from how much the muscle is stretched, you don't actually have an exact sense of how much your muscles are stretched, but you have this sense of what space you occupy, where your limbs are. And this sense is very important for simple tasks such as standing up, walking, but as shown in this picture, if you're very talented and practice hard, you can do things like that, thanks to your sense of proprioception. So, does this sense also depend on piezo2? And the answer is yes. In mice, if we engineer mice a certain way, we can see that they are extremely uncoordinated. Look at the hind limbs. It doesn't know where it is. It doesn't know where to put it. And this is a classical sign of lack of proprioception. Below, we're just showing that mechanistically, if you stretch these muscles and record from single fibers, in black you can see wild-type animals have a strong response, this pressure response. But if you knock out piezo2, they don't. One of the interesting things that happened after our studies in mice is our colleagues and collaborators from the NIH actually identified humans who do not have functional piezo2. So they have mutations in their piezo2 gene so that they do not have this ion channel as well. And in a fascinating way, the consequences of this lack of piezo2 exactly models what we had shown in the mice. So these individuals don't sense touch and don't have proprioception. So here's a video of one of these individuals who doesn't have a functional piezo2. And what you will see is that this person is asked to do something that most of us can do very easily, which is just walk in a straight line. She's blindfolded and harnessed to make sure she doesn't hurt herself. As you will see, she has a very, very difficult job of doing this. So this is because, again, the lack of the piezo2 channel that she has. So how about pain? We know both from our mouse and human studies that acute pain, the hammer hitting your finger, is still normal in these individuals. There's actually another ion channel that we don't know its identity. We're still looking for it. But what we're really interested in is more in chronic pain. And one of the symptoms of chronic pain is called tactile allodynia. Two complicated words, but it's a very simple concept. It just simply means when touch becomes painful. So you will experience this, for example, after sunburn, where just wearing a shirt or touching your shoulder will be painful, or after injury. Interestingly, this also happens for people suffering from neuropathic pain, for example, where any type of touch becomes painful and is something that we don't have good medicines against. So we showed that all forms of tactile allodynia, despite acute pain, depends on piezo2 in mice. And then our collaborators did a similar experiment in humans as a proof of concept if this mechanism is conserved between animals and humans. And these individuals who don't have piezo2 thankfully volunteered themselves for this experiment to show this important possibility. So the experiment goes the following way. We add an irritant to a part of someone's arm. It's just as capsaicin, the active ingredient in chili pepper that gives the burning sensation. And if you apply that to the arm in one little square, for example, it becomes red and it becomes sensitive, and you'll have this tactile allodynia where just touching becomes painful. So they do laser Doppler imaging to show that both control individuals as well as individuals who don't have a functional piezo2 in the below are actually inflamed where capsaicin is applied. And then the experiment is very simple. The experimenter touches the two boxes A or B and asks the individual which one is painful. So all the control individuals can easily tell 100% that the box B where it got the capsaicin became painful. However, individuals without piezo2 have no idea. They say we can't sense anything. So you have to tell them you have to guess A or B, and they randomly guess and get it 50% right of the time. So this is an important experiment for us because many times you find something in an animal model such as mouse, but it doesn't translate to the human, so it won't be useful for future medicines. This experiment told us that indeed in humans, if you find a blocker for piezo2, it will actually help individuals who suffer from neuropathic pain, for example. So what I've told you till now is that piezo2 mainly plays an essential role in all of these senses that depend on pressure sensing, including touch, proprioception, clinically relevant form of pain. I didn't talk about it, but also mechanical itch. But over the last years, we've also expanded now these studies to other areas, specifically to sensing pressure from internal organs. So there's a whole other sensory system, in addition to the DRG neurons that I talked about, that innervate the lung, the heart, stomach, the gut. And they gather both chemical and physical stimuli, information from these organs. And again, your body uses this for several reasons. And some of these senses you're very aware of, some of them you're not. But over the years, in collaboration with others, we have shown that piezos play an essential role in, for example, sensing lung stretch and regulating breathing. There's a whole neuronal system of detecting blood pressure to regulate it, to make sure that your blood pressure stays constant as you sit down, get up, go for a run. And piezos are absolutely essential for this. And you're not even aware of these senses. You don't know that you're sensing blood pressure. But behind the scenes, your neurons do this. Something you might be very aware of is urination, of course, and this is bladder stretch. So when your bladder is full, you know this very well, and it's, of course, an important sense. And interestingly, individuals, again, who don't have piezo2 cannot sense this at all. So in addition to not having coordination and sense of touch, they actually have, as they grow up, have learned to schedule going to the restroom 2-3 times a day to avoid accidents. So we're learning a lot going back from human and mouse. I just want to make sure I make it clear that this is not all solved problems. There's a lot more to do in this area, and I just showed this slide to illustrate this. So if you eat a very large meal, like I've been doing here in Yerevan recently, you feel very full, and that's a very physical, mechanical sensation, isn't it? But most of the research on how much you eat, obesity, diabetes, is actually centered on chemical nutrient sensing. So I think this is just an example of, in the future, we're studying how stomach stretch contributes to eating, overeating, and all the pathological consequences that come from it. So till now, I've been telling you mainly about sensory neurons and how we sense our environment as well as our internal organs. But these molecules also made us start thinking that it is not just important for those cells, but many, many other cells. Many of the cells in your body experience, sense, and respond to these kind of pressures, and we're just starting to find the biology behind them. So for example, your blood vessels sense the blood flow, and this gives a mechanical shear stress, and this signal is very important for regulating blood pressure. Your muscles get stretched. These signals are used in a variety of ways. And this mechanical indentation that we initially studied for sense of touch also relevant for other cells, including red blood cells, for example. So I won't tell you about all these studies, but over the last few years, we have found the sister molecule, piezo-1, the one that we first found in these neuro2A cells, is playing very interesting roles in different areas of biology. For example, both in mice and in humans, if you don't have piezo-1, your blood vessels don't even form correctly, because we found that early during development, correct flow sensing is absolutely required for the development of blood vessels. The next is not our work, but other colleagues have found that piezo-1 in bone is absolutely required to regulate bone density. You might have heard that astronauts who are going to outer space for a long time come back and they have reduced bone density, which is a problem. So it seems like indirectly piezos are sensing pressure on bones, and this is absolutely required for bone health. We found roles of piezo-1 in skin and wound healing, but the next few slides before I end, I want to tell you about this unexpected role that we've also found for these mechanosensors in both red and white blood cells. You would say, why would blood cells experience pressure? Well, red blood cells get squeezed through capillaries, which is half their diameter. And we and others have found that this pressure sensing is so important for these blood cells to regulate their cell volume, because they actually have to get smaller as they squeeze through this blood vessel. Again, in work that I won't tell you, we found that humans who have a mutation in piezo-1 that causes too much signaling instead of no signaling actually have dehydrated red blood cells. And these dehydrated red blood cells, although cause anemia, they also provide an advantage. In the mouse, at least, we've shown that they are protective against malaria infection. This is very unexpected. I never thought pressure sensing would have anything to do with malaria, but this is what we found. Again, I won't tell you about the details of this study, but I will tell you in the next few slides what this study enabled. So when we found that in mice, that piezo-1 protects against malaria, the first question we asked is, are there mutations in areas that is endemic for malaria that have this kind of gain of function, too much piezo-1 mutations? And when we looked, we found a very interesting allele. Allele is just another name for one of these genes' specific mutation. So this shows the distribution of this gain of function allele throughout the world map. The wild type, the normal allele, is in this teal blue color, and then this new allele that we found is in the purple color. So as you will notice, the purple allele is almost not present at all in people of European descent, for example, but people of African descent, where malaria is endemic, there is a substantial amount of this allele present in the population. In Africa, as well as, for example, in African Americans. And it is present in very high amounts. So what we call this is a subtle gain of function allele, which means it's just increasing the activity a little bit. It doesn't cause disease by itself, but as I'll show you, it's telling us some very interesting things in the clinic. So just the back of the envelope calculation says that about 1 in 3 people of African ancestry are heterozygous for this mutation. So this kind of translates into almost 400 million people on the planet that carry one allele, and 40 million would be homozygous or would carry 2 alleles of this. So what are the consequences? Just like many other mutations, mutations are not necessarily all good and all bad. They have both advantages and disadvantages. As I mentioned to you, the RBC, red blood cell deformation, as an advantage protects against malaria. This needs to be proven still in humans. But it also has disadvantages. I mentioned anemia. In this one study that I will show one slide of the next, we've shown that another disadvantage is that it causes increased iron levels, which is not good for the body. And this is how we're doing these experiments. So the original disease that causes too much piezo-1 that I mentioned is very rare. It's called hereditary xerocytosis, And there are only about 130 people worldwide that have been shown to have this. So from this, we bring this information into a mouse model. So we make a mouse model of this mutation, and then can mechanistically understand what's going on. Why do they have anemia? How do they have iron overload? For example, we found that it is this activity in these white blood cells, macrophages, that causes this phenotype. But then I find this very exciting. Then we can go to this very subtle mutation. That is, instead of the extremely rare 100 people worldwide, where 400 million people have this disease. And now can ask questions, are the findings that we did clinically relevant? So Sean, my postdoc in my lab, did this experiment where he got blood cells from about 300 African-American healthy donors. And this first slide just shows the distribution of this allele. As we expected, I told you, about 1/3 of African-American population are expected to have this mutation. So you can see the normal wild-type allele in gray, and in green you see about 1/3 have this mutation, and in orange you have a handful of homozygous, which means they have 2 alleles of this mutation. And then Sean asked, what happens if we, within this population, look at people who have high iron levels? So these are 2 clinically relevant measurements of iron levels. And when we look at these individuals, you can see that it's much more prominent within the people who carry this allele, as the gray part of this pie chart shrinks, and more we see people who have this allele have this phenotype. And if we combine these two, which becomes a clinically important way to look at iron levels, we can see that almost everyone in this category has at least 1 allele of this mutation. So this is very exciting for us because this was completely unknown, but we've shown that carrying this gain-of-function allele makes an individual 12 times more likely to have iron overload. And so this will help physicians both genotype, characterize, and hopefully someday treat any consequences of having too much iron. But of course, these are the first experiments. Larger studies are required. But hopefully what I'm trying to show you is this power of using human genetics as well as in vivo mouse models that we find novel mechanisms of disease. Just in the last 2 slides, I will just tell you that these piezos are evolutionarily conserved. So it's not just in mammals like mice and humans, but as this diagram shows, it's present in a variety of animals, including worms and flies, unicellular organisms even, as well as plants. And if you were earlier, they made me do this plant experiment. I will show one last slide on the role of this ion channel in a plant called Reductionist Approach. So first you would ask, what is mechanosensational pressure sensing relevant for plants? They actually rely on this in a variety of ways. One of them is, as plants send roots into the soil and they experience a harder surface, the plant has to make a decision either to go right through it or to go around it. You can't do this without sensing pressure. So Alireza Musavi, postdoctoral fellow in my lab, found that plant piezo is expressed in the root tips of plants, just exactly where you would expect pressure sensors to be. And he showed that if you now make a mutation in piezo so that Arabidopsis does not have this ion channel, it actually has a very hard time penetrating hard surfaces. So on the left you can see the normal white-type plant. When you, instead of a soil, you put this harder gel surface, they can penetrate right through it. And the way it's focused, you don't see its roots. However, in the piezo mutation, when the same obstacle arises, they cannot penetrate it. They go over and around it. And again, the way it's focused, then you can see the roots are very visible. I just show this experiment just to highlight how amazing it is that evolution has used the same molecular machines to sense pressure for you to sense touch and for plant roots to sense how hard the soil surface is that they have to navigate. So I just want to end by thanking the people who have worked with me over the last 22 years to do this research. So this includes current lab members, alumni who trained with me, who started their own labs or work in the biotechnology industry, as well as collaborators from all over the world shown on the right. I just highlighted a few names whose work I specifically highlighted. This includes Bertrand Goss, who cloned piezo, Sean Ma, who did the piezo 1 blood work, Rose for some somatosensory work, and Ali Reza, who did the plant work. And it's of course very important to also thank the funding sources, which is essential for doing this kind of research. And this is just names. I wanted to show a picture of a lab reunion we had a few years back, and we're going to have another one this year, so I'm very excited to do that. Also wanted to show a couple of more slides. This I really like. This is actually, of course, a world map, but you can see each of these pins marks the location of the birthplace of all members of my lab. And it illustrates the point of how international effort science is and how proud we are for having this excellent international collaboration. You can see I'm the one who's born in Beirut, and I hope to add a pin from Armenia in the next few years. And I go back finally on this slide that you also showed in the introduction. Luca, my son, who's sitting right there. This is a very tender, touching moment, if you will. So I just wanted to share with you how I found out about the Nobel Prize. So they, in Stockholm, they meet the last minute in their afternoon, decide to make the final decision, and then they call the awardees. That time in the California time is 2 a.m. So they tried to call me, but they couldn't get a hold of me because my phone was on Do Not Disturb. And they left a few messages, and then I guess they started Googling Patapoutian, California, and they found a number in North Hollywood, and they called, and it was my 94-year-old father's number. Later on, the head of the committee member says that my dad was very upset at him for calling. But I think he understood what was going on, and he finally called me and told me that I should pay attention to the voicemails on my phone. And so we're still in bed, just waking up. Nancy, my wife, went and got Luca, and the laptop, this laptop, is open just to moments later see the actual announcement being streamed live. So it was a very special moment, and because during COVID things have changed a lot, and at this time normally as soon as you get this news, hundreds of reporters come to your house, but none of that was happening this year. And this picture through Twitter made the news around the world and enabled me to connect to people all over the world, including these important connections that I've made and visited here in Armenia. I think, part of the story of this is not about me and my colleagues, but the fact that I'm of Armenian origin, and I'm very proud of this, and I hope it's an example to all the especially young scientists here that really anything is possible, because if it happened to me, it could happen to you. But most importantly, thank you so much for this warm welcome, and thank you for your attention.